we tried to data mine the patient claim database in the United States called the CDM. It has access to over 60 million patient records data in the United States. And what we're interested in is to identify, to see whether we could use this to systematically identify skin disease comorbidities when compared with the control. Okay, so we first look into around like 13 million individuals that have some form of skin disease, and then for each of those we control for the different potential confounding factors, and then compare the rate of comorbidities when compared with the controls. So this is like table to describe what we have found for those individuals that have at least some psoriasis diagnosis. So there are like over 270,000 of patients that we've identified. And then you could see the, see the mouse? Okay. So see there's like a breakdown of it. So around 25% of those are non-white, okay? So when comparing with many of those studies conducted in US, like for basic genetic or the molecular studies, which is Caucasian-based, there's still a substantial amount of patients here in here that's non-white. So we also have this like socioeconomic status that being shown here. We also show like the control breakdown. We look into the education level, household income. We see like a mild association. So this all suggests that when we really try to look into this kind of like epidemiological study, which have been described before, we really need to take this into account in the analysis. So this is a heat map showing the intensity of the odd ratios that were identified for psoriasis and psoriatic arthritis here. So this is like just some of the list that we show in here. Red color representing higher than expected, blue color representing lower than expected. You can see there's like multiple different comorbidities that we are able to identify. And when you look deeper into it, okay, so many of those actually have been described previously in different epidemiological studies. And it shows that how we could indeed using this database to like systematically identify psoriasis comorbidities. So we look into this a little bit more. Okay, so psoriasis type 2 diabetes association have been described previously in different studies, and I'm just showing two of the more recent ones that show the odd ratio between psoriasis and type 2 diabetes. It's around 1.2. So this study has like breakdown into different severity of the body size affected area. So and then we could by using this CDM database, we could actually track back to the last 20 years. Look at this trend. So here's what we estimated in here. So I know there's like a lot of factors, that come in here, like including ICD-9 to 10 changes, right? And also there's like more and more accumulated, like electronic health records data that are being available. So we broke down into like every 5 years in interval, okay? So we observe like relative consistent, stable odd ratio. We also see how the one that we estimated, like kind of aligning with what have been previously reported. We see a narrower credible intervals, as I mentioned, like because more recently we got more and more data, so we got a better estimation of that value. So I mean that like still questions arise, okay, including like, so in the past 10 years or so, there's like basics, science, research, translation research have been helping us to identify new psoriasis treatment options. But we look in the future, okay, with this success in psoriasis management, help us, to also look into a different trend of psoriasis comorbidities, right? And also how we could really optimize the treatment option for treating patients with comorbidities, okay? So with that, I want to introduce to the second part of my talk. So we're trying to really leveraging the genetics, focusing on genetics to see how we could identify genetic signals shared between psoriasis and its comorbidities. So this is like some of the comorbidities that you probably get really familiar with, and then many of those actually are complex conditions, indicating they are having like a polygenic nature. Okay, and then because of that, many of those genome-wide association studies have been conducted. Okay, so the remaining part of my talk is really trying to see how we could leverage this and compare with what we have from psoriasis genetic cohort. So, as described, so type 2 diabetes among one of the first psoriasis comorbidities. Okay, previous data show how more severe form of psoriasis will have greater risk of type 2 diabetes. Our goal is really trying to compare what we have from psoriasis against what have been described in type 2 diabetes to identify the shared and potential causal relationship. So this is like the cohort that we use. So psoriasis, we have like over 10,000 cases, and then in this type 2 diabetes cohort, we have like over 70,000 cases. And then this study is actually adjusted for the BMI, okay? And then we compare and then identify signals that are shared, within the same region, within the same genetic regions. So we identified 4 shared signals. Actually, interestingly, they are involved, seem to be involved in the NF-κB signaling pathway and also in the MAP kinase regulation. Okay, one of them in here is actually a missense mutation for the CHUK gene that activate NF-κB, and the rest actually have play regulatory roles, similar to many of those complex traits that does play a role in gene regulation. Okay, and then they are regulating genes in like STAT3 that play a role in NF-κB, and also the other 2 genes that in MAP kinase. So just an example. So this is like a regional association plot, each dot representing 1 genetic variant. Okay, you can see how the genetic signal for psoriasis is like in here, and then it's also comparing with that with the type 2 diabetes. So it's like currently in here, just in the same region. Okay, and then if you look into like a trans-disease meta-analysis, you can see the statistical significance got boops up, okay, and then you get like much larger association results. Okay. All right. So first, for previous introduction about data randomization. So this is actually a statistical technique, statistical genetic technique that we use. The idea is that when using the disease incidence, it could be confounded. By different confounding factors when inferring the causal relationship. So instead of that, we use the genetic signals to really model, the risk for the exposure, okay, and then in turn use that to model, the association with the outcome, okay, and then using that to make an inference about the causal relationship between 2 traits. So there are like multiple different assumptions that we have to make, okay, including the SNPs cannot have like independent association directly, okay, with the disease. So if that is the way we have to control for it, like if there's like potential pleiotropic effect with control for it, there should also be no association with some confounding factors. If there is, then we also have to control for it, like in like a multivariable model, okay. So we apply this to assess the causal relationship between psoriasis and type 2 diabetes. Okay, we only find statistically significant results for showing how there's a causal relationship from psoriasis to type 2 diabetes, but not the other way around. Okay, so we also look into like different model to try to control for potential pleiotropic effect or small effect sizes, and this result still stay the same. Okay. So we know that BMI is one of the confounding factors. And then one of our previous studies that actually show how BMI could have significant causal effect on psoriasis. And also BMI has been shown to be associated with type 2 diabetes, of course. So we look into a couple of things to make sure this is not, to understand, to show that this is like this causal relationship that we identify is not confounded by BMI. Okay, so first we look into the 4 shared loci. Okay, so only one of them is actually a BMI locus, and actually this is like just mildly having a linkage disequilibrium with the signal that we have identified, and actually the direction is actually the opposite. Okay, so indicating the 4 loci that we identify is actually indeed BMI independent. Okay, so and then again we look into like a BMI adjusted type 2 diabetes GWAS when we conducted our association, okay, And then finally, we also take into account the BMI GWAS signals, okay, in our causal relationship association. Okay, so this is like a result. Okay, so we put these two in the model to assess their causal association with type 2 diabetes. So you can see, BMI has a significant association with type 2 diabetes, like this the odds ratio. And even after adjusting for the BMI, you still see significant results, indicating that the causal relationship between, like from psoriasis to type 2 diabetes, is BMI independent. Okay, and we adjust for that. So we look into different disease comorbidities. Like in addition to type 2 diabetes, I mentioned also BMI. Okay. And we look into like coronary artery disease and multiple sclerosis, okay? And then we do identify, different causal relationships in some of our recent studies, okay? So I want to kind of look more into it, okay? So, just for example, multiple sclerosis and psoriasis, that's published earlier this year, we show like over 20 trans-disease signals between multiple sclerosis and psoriasis. So this is just like a circus plot, oops, sorry. To indicate, so this is like the signals that are shared, are in this panel here. The signals that are actually, opposite direction in the opposite panel, and then we actually show the different color representatives could be independent signals or not. Okay, when you actually look into the genes that that are encompassed in these regions, you actually see a lot of genes actually get involved in the IL-17. Or TNF signaling, or the JAK-STAT signaling, including some of those like really prominent genes that we have identified, like TNF-IP3, TYK2, TNF-RSF1A. Okay, so similarly, we conduct a Mendelian randomization to look into causal relationship, and we actually only identify causal relationship from psoriasis to multiple sclerosis, and you show here, and these results remain even if we look into like multi-variable model. Okay. Just some of the other factors that have causal relationship to multiple sclerosis, including like diabetes and also BMI, as like also low vitamin D levels, which is actually have been shown previously as well. So even after adjusting all this, like the causal relationship from psoriasis to multiple sclerosis still retain. Okay. So we look into like shared loci, whether they are asking whether they're in the same regions or not. So are we really talking about the same signal, or just like they just accidentally like reside in the same region or not? So we want to look into that using this approach called co-localization. So this approach has been actually used in interpreting some of the GWAS signals, okay? As I mentioned, like a lot of those GWAS signals are regulatory in nature, okay? So what they've been doing is like try to look into a potential region of genome associations, look into that, again, each dot represents one slip, and then look into potential gene regulation signals. So for example, this is like gene, this could be one gene, and then showing how each of those genetic variants is associated with gene signals, and then to see whether there's any alignment between them. So it's this cartoon just to show that, well, if there's a alignment between the GWAS signals and the EQTL signals, they would be like kind of overlapping really nicely. If not, even though they are reside in the same region, they are not co-localized. So this would help us to see whether, things within the same region are in the same signal or not, okay? So instead of using, GWAS and EQTL, we actually compare the genetic signals in one trait and the other trait, like type 2 diabetes or psoriasis. And I'm using like coronary artery disease and psoriasis as an example. We identify 4 shared loci between psoriasis and CAD, okay? And then we ask, there's like multiple different hypotheses that we test, and then each of those we compute like a posterior probability to see whether these signals are co-localized or not. So, among 4 of them, 3 of them actually have over 85% of posterior probability that show that they are indeed the same signal. For the last one, that actually have a lower probability. When we look deeper into it, there's actually like secondary signals, secondary genetic signals, encompassed within that region. And we control for these secondary signals, they're also co-localized. So this indicates that not only they are in the same region, but they are actually the same signal there. Okay. So this is actually just to show you what I mean. Okay, so this is like the regional association plot again in this chromosome 2. So it's actually, this is one of the signals in here, and then you can see how it's shaped, and it's actually having really similar shape when compared with that from psoriasis. And this is what we meant by whether they co-localized or not. So how about mechanism? So we talk about it in the same region, talk about there in the same signal. So what are potential mechanisms that involved? So we see that there's many of those are regulatory in nature. Some of those like a protein altering genetic variants. So we look into like a multi-omics data. So this is like a lot of information in here. So we look into whether the genetic. So first, okay. So when you look into it, so these are the 4 different loci, okay. And then for each locus, we look into the genes within that region, and we try to prioritize them, okay. So some of the features that we use to prioritize them, including whether the genetic variant is able to affect gene expression nearby, okay, whether there's actually like methylation associated variants, and also asking whether they express or not in the skin, so this is like also an example from our study in comparing the CAD and psoriasis. So are they actually expressed in some of those like heart tissue, the skin tissue, or there's actually missense mutation or not? So among 4 of these, 2 of the loci actually we are actually narrowed down to 1 gene. Okay, so this IFH1 is one of the really prominent genes that we have been previously identified psoriasis, and actually this particular signal, okay, that we identified to be shared signal between CAD and how this is actually have been reported in a singleton-Merten syndrome, okay, rare heart condition. And then the other one is actually pointing to a regulatory genetic variant, to a linked RNA. And this particular loci also have been associated with artery stiffness in a UK Biobank study. Okay, so this using multi-omic approach to actually help us to tease down some of those this same signal is like what kind of potential regulatory mechanisms they got involved in. So we talk about mechanisms, shared signals. So what's a potential translational implication when we are dealing with this like genetic data? Okay, so I'm using like predicting psoriatic arthritis as an example. So we're not able to include a lot of other like environmental variables as discussed. But here we're focusing on genetics. Okay, so the idea is that can we actually predict psoriatic arthritis among psoriatic patients? Okay, by only using genetic data alone. Okay, so this just one slide summary about what we have done. If we look into around 10,000 individuals. Okay, so some of those have like psoriatic arthritis status. Some of those have not been diagnosed with psoriatic arthritis in a 10-year period. Okay, so they're like cutaneous only psoriasis, and we're really trying to say, well, can we predict them? Can we separate them? Okay. And then we look into our data that have genome-wide coverage or not. Okay, so a conclusion is that we're able to achieve up to like 0.8 in area under ROC. And then using genome-wide data is actually much more informative than only focusing on regions that have been previously reported to be associated with the immune diseases. Okay, so this 0.8 is like okay, but it's not the best. So we look into the precision recall curve. So still, when we look into the top X%, like for example, top 10% of the patients that we predict to be psoriatic arthritis, we have over 90% accuracy. Still, this is only really small proportion of individuals, but we are only using genetic data. So as previously talks just discussed, when we integrated more and more information to take that into account, we hope it could improve this prediction better. So in summary, so the CLAM database have provided really good resource to assess a trend in psoriasis comorbidities. The previous study have shown like, we rely on this physician diagnosis codes only, codes only from the CLAM database could have good accuracy around over 80%, but still, I think there's like more information that we could leverage to have increase the accuracy in the diagnosis when we just look into the electronic health records data. So, and then by using the transient meta-analysis, also the different statistical genetic approaches, we could identify the shared genetic signals and their causal relationships. trying to actually account for the potential confounding factors. We show how integrating multi-omic information can facilitate inference about the shared mechanisms. And then when using machine learning approach and only applying to the genetic data, we're able to achieve satisfactory results. But can we really apply to independent cohorts? So it's something that we need, get together like multiple different centers information to achieve. So with that, I would like to thank all my colleagues, my mentors, and then also my groups here, especially Matthew Patrick, who have done a lot of great work in these genetic studies. Happy to take any questions. Thank you.
Thanks very much indeed, Alex. We've got time for one or maybe two burning questions.
Yes, please.
Mark.
I was just thinking, obviously with the other large efforts in other countries and the fact with EPIC being, how would you say compared to the opt-in data, if one was able to eventually get into, say, getting all the EPIC data together? Would there be any better, granularity, evolution, etc., that may provide more value, or at least additional value over what the opt-in database provides?
Right. So I guess the question is, I think first we have to make sure this could be synchronized, like kind of like having a way to synchronize the definition between the different cohort, right? So that's actually one of the really important points, And then it's always good to have this because you also want to test whether some of those results that you identify could be independently validated, right? But of course, some of the other things, including what's the ancestry compositions in some of these cohorts. When we show this in here, we try to take into account self-reported ethnicities. But some of those, like after we identify, that that association may not be really validated in other independent cohorts just because of different ancestry compositions. And also, there could be other things that you take into account, including, what's the sample size in some of those subcohorts. We are talking about in here gathering like thousands of different clinics, right? So some of those have really sparse data in each of the individual centers. So yeah, I think I would say obviously it's good. There's a short answer, but I think there's a lot of hard work that we have to try to synchronize this kind of data. Yeah. Yes, please.
Very quick question, very quick answer. Andy.
So, chronic inflammation, a bunch of things that have chronic inflammation are associated with heart disease, right? So not just psoriasis, but gum disease, so forth.
Yeah.
So when you publish that, again, I'm asking you again, so you can say it publicly, that heart disease predisposed to psoriasis, it just seemed to put everything on its head for me. So
yeah.
I mean, are you saying that chronic inflammation does not lead to heart disease?
So okay.
The short answer.
Okay.
It's not that.
Yeah. So we are trying to marginalize this. So one thing that I want to just mention this is that there could be late onset of psoriasis. So we may be talking about potential subclinical inflammation in the heart that lead to the later onset of psoriasis. So yeah, but I could talk about a long answer.
Something for lunchtime.
Yeah. Yeah.
Thanks very much, Alex.